Hi all, thanks so much for joining today. We're going to be starting the seminar promptly at noon Eastern time, so in 5 minutes. Hi everyone, thank you all so much for joining us today. We're going to be starting promptly at noon Eastern time, so in just a few minutes. All right, we're going to go ahead and get started. So hi everyone, thank you all so much for joining today. Welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I am the NOAA OAR Omics Coordinator, and I sit in NOAA Ocean Exploration. I also sit on the NOAA Omics Working Group as the Executive Secretary and as a subcommittee co-lead. So omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA and also with our NOAA partners. So the seminars from this series are all made available on YouTube and then posted up on our omics website. There should be a link to our omics website in the chat now if you'd like to go back and look at our past seminars or register for the next seminar. So as participants, you all are muted, but feel free to type questions into the questions for staff box throughout the presentation or at the end of the presentation. And at the end, I will read the questions for our presenter to answer. All right, so with that, our presentation today is titled Using Metabarcoding to Understand Microbial Communities on Aquacultured Sugar Kelp, Identifying the Good and the Bad Players by Dr. Yuan Liu. So Yuan, I'm going to go ahead and give you screen sharing permissions. So let's do that. And while Yuan is getting set up, I'm going to introduce her to our audience. So Dr. Yuan Liu is a marine molecular biologist. She earned her PhD in microbial oceanography from Stony Brook University, where she used a DNA fingerprinting technique to study planktonic microbial communities associated with a harmful brown tide species. Working in the NOAA Northeast Fisheries Science Center Milford Laboratory, Yuan strives to apply environmental DNA, or eDNA metabarcoding, to a variety of fisheries-related projects that examine all kingdoms of life. Currently, her focus is to contribute to the NOAA Omics Strategic Initiative by identifying best practices for eDNA surveys that could be added to the agency's fisheries survey toolbox. And Yuan, I know we very much appreciate your participation on our NOAA Omics subcommittees and contributing towards the strategic plan and strategy. So with that, I'm going to pass it off to Yuan. So Yuan, I can see your presentation. Do you want to come off mute and just do a quick audio check?
Absolutely. Thank you, Catherine. Could you hear me okay?
Yep, we can hear you great.
Thank you very much. Thank you for the very nice introduction, and thank you for inviting me, Catherine, to speak at this webinar series. Welcome everyone to this talk where I'm going to share our work done on the microbial community on aquacultured sugar kelp, Sucrina latissima. Oh, need to move my cursor so it can move. All right, so before I get started, I would like to first acknowledge my co-authors at Northeast Fisheries Science Center at Milford Laboratory. As a group, we did Vibrio culturing and visual inspection, microscopic identification, and of course, metabarcoding. And Lina Donnarumma assisted me with the lab work, with the eDNA lab work. And our industrial partner, Greenwave, helped us identify a sugar kelp farm and also assisted us with the field sample collection. Last but not the least, NOAA Fisheries Office of Aquaculture and NOAA Fisheries Omics Strategic Initiative provided the needed funding to complete this project. Just a little bit on the background, why do we care about sugar kelp? As a winter crop, sugar kelp is a large brown macroalga that takes advantage of the increased nitrogen availability in the colder months of the year when other aquaculture activities are at a minimum. It's rich in multiple nutrients, therefore it's considered the next kale. Its Asian cousin, I guess, Sucrina japonica, is a pretty popular food item in many Asian countries. And here in the United States, based on my personal experience, I think you're more likely to find sugar kelp in salad or soup in some of the ethnic restaurants. So I personally have read many reports on using sugar kelp to make beer, including this one. I hope you can see my cursor okay, from NPR. And many researchers have also started looking into aquaculture sugar kelp's potential to mitigate climate change and to serve as a global energy source for the future. I think it's fair to say, even though we're still cultivating and exploring market interests for aquacultured sugar kelp, its aquaculture is taking off in the United States. So surrounding the challenges about its aquaculture sugar kelp is generally considered as safe when used only as a spice by FDA. This is to say, if we want to use it more than as a spice, there's a lot for us to learn about its potential food safety hazards. And then from a local perspective, in Connecticut, where I am, sugar kelp farming follows the same guidelines used for oysters, which is normally considered as over-restrictive. This is why we did all this work, to better understand the epibionts of aquacultured sugar kelp that are important for human health and also for kelp health, especially the understudied molecular components, microbial components. Ultimately, we would like to use our work to help enhance the regulatory framework for kelp farming. So before we started collecting field samples, we had some specific, like targeted questions and some more exploratory questions. A very clear direction for us to start with, of course, is were there any hazardous microorganisms on sugar kelp? And this for human pathogens include pathogenic Vibrio, specifically Vibrio parahaemolyticus and Vibrio vulnificus. And again, this is based on what we already know about oysters. These pathogenic vibrios can proliferate in oysters, so they are certainly one of our targets. And then based on what is already known about Long Island Sound, what has been detected within Long Island Sound, Lyngbya, a cyanobacterium, and Prorocentrum lima, a dinoflagellate, are both toxigenic and also epibenthic, and therefore were also our target. And then the question on what the possible kelp pathogen could be is more of an exploratory question. It's more exploratory because there's not much information that's available for us to consult with. And naturally, if we find any microorganisms from the first question that seem to be interesting, then of course we would like to follow up and see what their temporal patterns are throughout the kelp growth season. And lastly, in microbioceanography, there's a well-accepted notion: everything is everywhere, and but the environment selects. So I think in this case, it's kind of reasonable to expect kelp surface to function as a selecting agent to form and to shape its own microbial community. So I was very interested to see if the blade and seawater microbial communities would differ and what the differences could imply. This is a quick slide just to show you the methods we use to target the visible and the invisible. Like I've mentioned, Vibrio is one important group as our target. So we use TCBS agar to enrich the growth of Vibrio strains. And of course, not all Vibrio bacteria are pathogenic. So we use PCR screening for the ToxR gene to decide the presence of the pathogenic Vibrios. We use light microscopy to identify microalgae on kelp plates. And then the visual observations were also helpful to us to identify some of the organisms that might compromise kelp growth. For example, lacy bryozoan, they can actually form colonies on kelp plates and compromise its integrity. And snails, for example, northern lacuna, on the other hand, are active kelp grazers. Lastly, the focus of this talk is going to be the metabarcoding work. That is the partial sequencing of the 18S and 16S ribosomal RNA genes, which will target both the prokaryotic and then the eukaryotic microbial communities. So with that, this slide shows the sampling map. The kelp farm is located on the eastern end of Long Island Sound at Groton, Connecticut, on the eastern end. Eastern end is the relatively open end, while the western end of Long Island Sound is probably better known for its seasonal hypoxic conditions. The sampling took place in year 2018 and it covered 2 months, April and May, the major growth season of kelp. And the sampling frequency was roughly biweekly. And at each time point, 3 blade samples and 3 seawater samples were taken for metabarcoding analyses. Kelp looked pretty pristine throughout the whole sampling season, until at the very last day, May the 30th, as you can see on the tip there was some light biofouling on kelp. I will say I'm most familiar with like processing water samples. So when it came to biofilm samples, I decided to do some testing to make sure I would be using an effective DNA collection method. And to do that, I actually acquired some samples in the year prior to the field study, in year 2017, and in the month of July. I chose July samples because there was quite visible biofouling at that time. Therefore, I knew there would be enough material for me to work with. And I tested 3 collection methods on 3 parts of the kelp. One is the tip, that's the oldest growth, and then intermediate part, that's the kind of intermediate growth, and the part that's closest to the holdfast, that's the newest growth. So I'm going to show you that result first. Well, I ran out of space in the paper, so I had to push this part into the supplementary material. But personally, I was pretty impressed by how different the 3 collection methodologies turned out to be and how obvious the best choice should be, which in my experience is kind of rare when it comes to DNA method comparisons. And I'm going to just like tell you very quickly what I did in the 3 methods. In method 1, I used a brush to kind of brush off the surface content from the kelp and while applying a steady stream of sterile seawater to collect the surface content. And the surface content within the sterile seawater was used to filter through 0.2 micron polycarbonate membrane filters. And then the filters were, of course, used for DNA extraction using QIAGEN's Power Water Kit. In method 2, I used a sterile cotton swab to scrape off the surface content from the kelp. And then the cotton swab was used for DNA extraction using QIAGEN Power Water Kit. In method 3, I also used a cotton swab to scrape the surface content off, but different from the previous 2 methods, I used QIAGEN's Power Biofilm Kit to extract the DNA. You can see from the results here, the best method combination was definitely brush and power water combination, both in terms of the total DNA yield and in terms of the pattern that was captured. The oldest part of the kelp had the greatest DNA amount per area. So clearly that was the method that we chose in the following year, 2018, when we did the field work. So moving on to 2018, that's kind of the core part of the results. First, I'm going to show you this class/phylum view of the 16S and 18S community composition. So the x-axis is generally time, and y-axis is relative abundance. So I made the plot so that x-axis on the x-axis, samples collected from blade and seawater samples collected from the same time are next to each other. So for example, as you can see here, this is T1B, this is T1W. So that's time 1 blade and time 1 seawater. So they're next to each other. And then we move on to time 2 and time 3 and so on and so forth. It's kind of difficult to appreciate details, but if we can spend just a few seconds, we can probably see some differences between different groups. I'll just point out a few groups here. In the 16S community, the relative abundance of this red bar actinobacteria was greater in seawater samples than on the blade samples. And the opposite, I guess, can be said for this color, dark orange, which represents Gammaproteobacteria, who had greater relative abundance on blade than in seawater. And then if you look at a couple microalgae groups in the 18S community, let's look at this blue bar first. This is Bacillary-Phyta, which are diatoms, and diatoms had generally greater abundance on blades than in seawater. The other microalga group, dinoflagellate, represented by this purple bar, which here, if you can see my mouse, had greater relative abundance in seawater than on the blades. So clearly, this kind of high-level examination of data is helpful because you can pretty quickly say the blade samples look different from the seawater samples by just focusing on some particular taxa. However, I want to know if such differences can be observed on the community level. And if indeed the blade communities are different from seawater communities, what microorganisms were driving the differences, or like were making the blade samples blade and seawater samples seawater. So I'm going to show you a slide that shows some of my thought processes that led to the statistical analysis done in an attempt to answer these questions. So the first question I asked, of course, is, was there any structure in the data matrices? And that's the community difference I was talking about. And to answer that question, I used ordination analysis, NMS, non-metric multidimensional scaling. And the answer was actually yes. So I went on to define groups and used PermaNoVA to see if the predefined groups were indeed significantly different. How I defined the groups was based on both the ordination pattern observed in NMS analysis and also the intrinsic design of this experiment. Because you may remember there are two pretty important factors. One is, let's just call it site. That is the difference between the seawater and blade. And then the other one is sampling time. And again, PermaNoVA told me, oh, these factors were indeed important. At this point, I was most interested in what microorganisms were driving the site difference. And to answer that question, I used indicator species analysis, ISA. So indicator species analysis was conducted on data matrices of read numbers of molecular operational taxonomic units. And for the rest of the talk, for easiness, I'm just going to use MOTA to represent molecular operational taxonomic units. What ISA does essentially is it looks for the most consistently abundant motifs on blades and in seawater. So you can imagine a perfect indicator species would be appearing in only one group. Therefore, blade indicator species and seawater indicator species were the microorganisms that were mostly enriched on blades and in seawater. So a little bit on the work that I did, like putting 16S and 18S communities together. I tried a little bit of the network analysis. Suffice to say, I decided I was just going to use mental tests to decide what are the most important associations between the 16S and 18S communities. Now I'm going to show you the results along this way. Let's start from the ordination analysis first. So this is the NMS ordination pattern of both 16S and 18S communities. Here, let's look at 16S community first. So the ordination solution is one-dimensional, which means that one axis was sufficient to describe the pattern in bacterial community. And in this case, 81% of the variation in the original data was explained, was represented by this solution. And you can see here, the most striking structure, I would argue, was the separation between blade and seawater bacteria. And for the blade bacteria, they demonstrated some temporal variation, which was not the case at all for the seawater bacteria. So if you now look at the 18S communities, the solution in this case was two-dimensional. And still, the major structure in the 18S community, which kind of explains 76% of the variation in the original data set, was the separation between blade and seawater eukaryotes. And a minor structure, about 15% of the variation in the original data set, was this temporal separation for both blade and seawater eukaryotes. So to put the 16S and 18S data set together, I hope you can agree with me that the major difference is the site separation. Blade samples looked very different from seawater samples. In terms of the temporal separation, it seems like blade samples demonstrated greater variation, although again, temporal separation is a minor structure compared to the site separation. Seeing that the site was the most important factor and sampling time may also have played a role, in the next step, I set up PermaNoVA as a two-factor analysis for site controlling for sampling time. So now let's look at the PermaNoVA results. If you look at this column of p-values, as you can see, both site and time were indeed significant. So was interaction term. So this is to say there's a lot to the data. And at this point, I decided my focus was going to be the site difference, which was also backed up by the F values. As you can see, both 16S and 18S community F value for site was the greatest. So then now we move on to the next question for site. What microorganisms were driving the site differences? And here is when I use the indicator species analysis. First, let's look at the prokaryotic community, the 16S community. The upper panel, we see the blade indicator species, and the lower panel, we see the seawater indicator species. If you see these two numbers, 20 versus 74, we had many fewer 16S motus that were identified as blade indicator species than seawater indicator species. This was the first time it hit me, I wonder if we can kind of interpret this as blade being a more suitable habitat for a relatively small group of bacteria, more specialized bacteria who did better on surface. Now, the next, I'm just going to show you some groups that I think were really interesting. Of course, I said our focus is going to be on the site difference. And if you look at this blade, this large pie, which was Gammaproteobacteria. Gammaproteobacteria actually contributed more than half of the blade indicator motus. And among them, there are kind of a couple good players, potentially good players, Marinobacter and Alcanivorax. So these two bacteria actually have been established to play important roles in oil remediation projects in marine environments. Our identification of these bacteria as blade indicator species kind of prompt a question, can we use aquacultured sugar kelp in oil contaminated environments, marine environments? I also identified some potential bad players, specifically potential kelp pathogens, based on two criteria. First, these bacteria have to be doing really well on blades. That is, they have to be blade indicator species. And second, there has to be existing evidence to show that they actually infect macroalgae. And Aquamarina actually meet both criteria. Aquamarina is a flavobacteria genus that has a lot of agrolytic and chitin-degrading strains that cause eukaryote diseases. For example, bleaching in commercially important red macroalga nori. That's the sushi seaweed. So its association, Aquamarina's association with brown algae, which again, I will remind us, the sugar kelp is kind of brown algae. Its association with brown algae has only recently been established, but we do not yet know if Aquamarina is agrolytic towards brown algae. And then another group of bacteria, Parcubacteria, was another group that I labeled as potential kelp pathogen. So this is a candidate phylum that was unique to blade samples. And so far, no Parcubacteria strains have been isolated in laboratories, and all Parcubacteria sequences have been isolated from anoxic conditions. A recent publication actually discovered Parcubacteria have significantly reduced genome size, linking to fermentation lifestyle and the absence of biosynthetic pathways, all pointing to a very special kind of lifestyle, including parasitism. So this is why I label this Parcubacteria as a potential kelp pathogen. So in terms of the seawater indicator species, I'm not going to say much about it, but I would just point out many of them are known abundant marine heterotrophic bacteria, for example, Alphabacteria, Pelagibacter. So, of course, the two kind of interesting groups we found as potential kelp pathogens are Parcubacteria and Aquamarina. And then if you look at the temporal pattern of these two genera, I mean, first, I guess there's no synchronization in the growth pattern of these two bacteria throughout the kelp growth season. However, look at like Parcubacteria in mid-April, there was a peak. It's really interesting in, mid-April, kelp looked pristine and there was no sign of anoxic conditions. So it's really interesting. What does this mean? Maybe there are Parcobacteria that are not anoxic. What exactly were they doing in mid of April? I think this is a very interesting group to kind of have some follow-up studies on. And now we move on to the 18S indicator species. Again, upper panel is blade and lower panel is seawater indicator, 18S indicator species. Similar to 16S communities, we had many fewer motus, 18S motus that were identified as blade indicator species than seawater indicator species. And if we look at the specifics of the groups, I would just say from 18S indicator species analysis, it's really amazing how this analysis was able to kind of identify blade and seawater indicator species whose ecology are very well aligned with what we know about these microorganisms' ecology. And I will show some examples to show what I mean. So Bacillary-phyta, again, diatoms. We identified motus that were blade indicator species and seawater indicator species of diatom motus. And among the blade indicator diatoms, beside 5 that were unclassified, all the rest 9 classified were indeed benthic genera. On the other hand, seawater indicator diatoms, all 5 modules were indeed planktonic. Another group is ciliates. And blade indicator ciliates, actually the class Phylopharangia specifically, is known to include a lot of species that are morphologically adapted to benthic lifestyle. And then on the other hand, the seawater indicator ciliates, and actually only these 2 genera specifically, Spirotontonia and Strombidium, are mostly planktonic. So I think that was pretty interesting. And then based on the indicator species analysis, I also labeled another group, Peronosporomycetes, as a potential kelp pathogen. Peronosporomycetes is fungus-like, yet taxonomically stromatophiles. They were actually unique to blade samples. They are known to be a major pathogens to red microalga, again, the commercially important species nori, and brown microalga, giant kelp. Considering their commercial significance, it's kind of amazing how little we know about this eukaryotic group. So the only effective treatment to disease nori, as we know now, has been to acid wash the crop. Based on what we have found here, I think using more specific primer set, especially ITS primers, will be very helpful to help us identify the specific strains that are associated with aquacultured sugar kelp. Challenge experiments where aquaculture sugar kelp can be exposed to pathogenic strains of Peronosporomycetes will also be very helpful to help us understand what exactly they do on the kelp surface. I think that's pretty much what I want to say for this. And actually one last point on this previous slide. As you can see here, I also labeled the blade indicator ciliates class Phylopharangia specifically as good, and I am going to show you why I think so on the next slide. So this is a temporal pattern of the 18S blade indicator species. On the left column, we see epibiotic, the epiphytes, the blade indicator epiphytes, including epibiotic red algae, Rhodophyta, epibiotic brown algae, Phaeophyta, and also epibiotic diatoms. And on the right column, we see blade indicator heterotrophs, including the potential kelp pathogen, Peronosporomycetes, and 2 grazer groups, arthropods and ciliates, again, Phylopharangia. So if I'm just going to first point out the temporal patterns of, on the y-axis actually are the read numbers of these different groups. Let's look at the temporal patterns. The temporal patterns of the epibiotic red algae and brown algae were really similar. As you can see here, they gradually increased in the read number from T1 to T3. That's throughout the month of April, and had this significant dip in their read abundance at T4 and had a recovery at T5. Epibiotic diatoms actually, looked similar in that it had a significant dip at T4 and had a recovery at T5. But what was different for epibiotic diatoms from the red algae and brown algae was the read abundance for diatom was already pretty high and stayed high throughout the month of April. And I will say the potential kelp pathogen Prorocentrum followed really similar trend as epibiotic, especially red algae and brown algae, in that there was an increase from T1 to T3, that's throughout the month of April, significant dip at T4, and a recovery at T5. So if you look at the ciliate group, Phyllophrangia temporal pattern from T1 to T4, there is a very steady increase in its read abundance. And this is a significant peak here. What I think this temporal pattern shows is actually some really interesting trophic dynamics. And I'm going to explain myself and see if you can agree with me. So let's start from the epibiotic diatoms. So throughout April, the abundance of diatoms seems to decrease a little bit, which aligns well with the general understanding of diatoms being an early spring bloom species. At the same time, throughout the month of April, the abundance of epibiotic red algae and brown algae increased. Throughout the month of April, all this growth of epibiotic, epiphytes did not cause appreciable biofouling, likely attributable to active grazing from the ciliate group Phyllophrangia. As a matter of fact, like I've already shown you, the abundance of Phyllophrangia actually increased from T1 to T4, probably benefiting from increased preabundance, including diatom and other epiphytes, who I believe themselves were benefiting from the condition of like increased daylight and also increased water temperature to photosynthesize more. I believe that Perennosporomycetes probably experienced similar grazing pressure from Phyllophrangia because it followed a pretty similar temporal trend, especially to red algae and brown algae. This last, like at T5, this like significant jump in the read number of arthropods was really interesting. It probably reflects a transition from the dominant grazing group from Phyllophrangia to arthropods. Unfortunately, the primer set I used to identify 18S members was not good enough and was only able to tell me to the class level. This arthropod motu was actually a group of Maxillopoda, which includes copepods. So, we all know copepods actually do graze upon ciliates. So if this arthropod motu was indeed some kind of a copepods, so what may have happened was as ciliates, Phyllophrangia was grazed down by copepods at T5, the rest of the community, the blade indicator epiphytes and Perennosporomycetes were kind of enjoying this kind of grazing pressure, like a lack of grazing pressure from ciliates due to this trophic cascade effect. Thus, this small recovery at T5. So lastly, I just want to show you the Muntel tests that I did trying to combine 16S community and 18S community. I computed the Muntel tests of all the 6 possible pairs of the 4 data matrices representing blade prokaryotes, blade eukaryotes, seawater eukaryotes, and seawater prokaryotes. There were only 2 associations that were considered significant, and that was between the prokaryotes and eukaryotes on blade, and between the prokaryotes and eukaryotes in seawater. No significant correlation was found between seawater and blade microbial communities. Without extrapolate too much, I think what this may suggest is that the most significant biointeractions between microbial communities was within the same habitat. So I will say the kind of the greatest challenges for me in wrapping up this project was to corroborate results from using different methodologies, for which metabarcoding is only one of them. Results from 18S and 16S communities, and also results from seawater and the surface of the kelp. So I hope the paper offers some, more information on how different components connect. And here I'm just going to point out 4 major points as a conclusion slide to this project. So first, we did not detect any pathogenic Vibrio. A Vibrio mode2 was indeed detected using metabarcoding, but its abundance was extremely low. Neither Lyngbya nor Prorocentrum was detected either. And Parcobacteria and Aquamarina in 16S and Peronosporomycetes in 18S were detected as potential kelp pathogens. And combining metabarcoding and indicator species analyses, we were able to identify some potentially important trophic dynamics. Grazing from the ciliate class, Phylopharangia specifically, may be important in keeping epiphytes and a potential kelp pathogen in check. And lastly, the significantly enhanced growth of L. carnivorax and Marinobacter may suggest that aquaculture sugar kelp can be useful in oil remediation projects in marine environment. So this is going to be my last slide. Like I quote unquote promised at the beginning of my talk, I will want to kind of talk a little bit about the differences and similarities between eDNA from microorganisms and macroorganisms. If you will allow me to use a kind of dichotomy view of to approach this, we have traditional and we have eDNA methods to study microorganisms and macroorganisms. So the solid lines represent the kind of better established or more often used method in contemporary publications. And that is, for microorganisms, we usually publish using metabarcoding. And then for macroorganisms, for example, for fish, we use trawling, or we trust trawling data more. And then for the less established method, of course, we have we have something like light microscopy to study microorganisms, and we use metabarcoding to study macroorganisms like fish. Because I study fish eDNA right now, so I'm just going to use fish to represent macroorganisms. You can think of your favorite animal or plants to substitute fish. This is not to say we don't have many publications using eDNA metabarcoding to study fish, but rather I feel like we still bear the responsibility to prove metabarcoding data's worthiness in studying macroorganisms. Therefore, it's the less established method. So what's my point here? I had hoped to do some method comparison between just to see what microorganisms, what microalgae we would detect using light microscopy and metabarcoding. Something of interest to me would be what species tend to be dropped or underestimated using microscopy, and what species tend to be underestimated by metabarcoding. That didn't really happen because we had limited resource and time to finish the project, but the fact it did not happen made me think. I will admit it's definitely not the most groundbreaking question in the world of microbiology to understand, to ask the question, what is the association between a method like metabarcoding and light microscopy? In the world of microbial ecology, I think it's pretty well accepted that we rely only on molecular data to derive information for microorganisms. In contrast to that, in the world of macroorganisms, like for fish, how traditional surveys like trawling compare with EDA metabarcoding is probably one of the most intriguing questions that we as a community would like to understand. Therefore, you're seeing this small question mark, maybe some interest, and this question mark, a lot of interests. The difference in the value we see between such comparisons definitely makes a lot of sense, but I do wonder if it is always bias-free when it comes to our perception of a preferred or better accepted methodology when studying bacteria versus fish, for example. So we didn't really do a systematic comparison between light microscopy and metabarcoding, but we did capture a lot of images for microalgae. So here we just like show some of the images we captured. Of course, we detected way more species using metabarcoding, which is kind of expected. However, even for the most abundant species, metabarcoding and light microscopy did not always agree. Here I show some examples. These kind of species in bold actually were species detected using both methods, and also they all seem to be abundant using both methods. On the other hand, these two species, these two diatom species, they were present, but there wasn't much agreement between the two methodologies we used. So the second point I want to make here really is the importance of repeated data mining for both micro and macro organismal eDNA data sets. So now working on fish eDNA, I care very much about differences between, say, 97% and a 100% match between query sequences and reference sequences. I care very much about what fishes look like and what they do. For microorganismal data sets, it's not like you don't care, but usually there are hundreds of thousands of motus with 100% match to reference sequences. Most of the time, however, these are naturally occurring microorganisms with no lab cultures. We know very little of what they do in marine ecosystems. So in this case, I think repeated data mining, both before and after publication, in my opinion, is extremely important in updating our knowledge of the microbial functions. Because as new information becomes available, it increases our ability to formulate and test specific hypotheses. And I want to share with you one example that I think can show the benefit of learning from each other's research. I listened to a talk by Dr. Kathy Pfister at University of Chicago and Dr. Jessica Mark Welch at MBL earlier this year. They used a sophisticated version of fish fluorescence in situ hybridization to look at the spatial location of different bacteria on bowl kelp. Among many other things, they found this Gammaproteobacteria genus Granulococcus to be commonly present on their kelp samples. I'm attaching an image here and also the reference in case you're interested in checking out that publication. So Granulococcus did not stand out in my indicator species analyses, but after I listened to Kathy and Jessica's talk, of course, I went back to check my original data set. Sure enough, they were present, they were there. I probably would never have paid attention to this group had I not tuned in to Kathy and Jessica's presentation. On the other hand, you may still remember I found, for example, Parahaemolyticus, the supposedly anaerobes unique to blade samples. So I reached out to Kathy and Jessica after their talk and learned that they did not design probes to target this group of bacteria. Quite understandably, because they are a candidate phylum, there's probably very little known about them. You can never really design a probe. You can't possibly design a probe if you don't know they're present in your samples. So selfishly, I hope some of the blade indicator species highlighted in this work, such as the 16S Parahaemolyticus, Aquamarina, the other potential kelp pathogen, and even the 18S Peronosporomycetes can be further investigated. So the last reflection point I want to share is a kind of potentially controversial one. and that is if the metabarcoding is quantitative. First, I'm just going to share my opinion. I've always believed that metabarcoding is more than just presence and absence. So I'm going to share with you a couple stories. For my dissertation work, I used a DNA fingerprinting technique to study a brown-tide species, Aureococcus anaphergeferens, and the rest of the associated microbial communities. So the fluorescence peak of the DNA fragment representing Aureococcus had a very strong correlation with Aureococcus counts using a monoclonal antibody technique. During my PhD defense, one of the community members asked, they were pleasantly surprised, but at the same time, they just asked, how can this be? What does this mean? It's a fair question to ask because the usually abundance indices derived from PCR processes, we take those with a grain of salt. Because there are a lot of factors that can kind of complicate the equation. For example, different species have different copy numbers, ribosomal genes, and also PCR process is intrinsically biased. So it's quite understandable that I was asked that question. I probably said something to the effect of, conventional PCR may be more quantitative than what we had thought it to be. Miraculously, they let me pass. I think it was 6 years later in the meeting held at Woods Hole, where I said in the presentation that I gave about EDM metabarcoding. I said that EDM metabarcoding has very quantitative information for at least major groups in the community. And I made that statement because I was basing that on the EDM metabarcoding work I had done in Land and Sound about fish. And basically, my data showed that the temporal and spatial patterns revealed about fish using EDM metabarcoding made sense. And we even found evidence of a species migrating northward. I definitely noticed at least a couple of the audiences disagreed with me at that time. Now, of course, I couldn't see your facial expressions. I'm just going to risk it and say the epibiotic and planktonic species identified as blade and seawater indicator species and the trophic interactions I think was captured in this study shows again that there is a very quantitative information from metabarcoding. We may not immediately be able to say how numbers relate to individual numbers, especially when it comes to macroorganisms. Although I do want to encourage those who are interested to check out Mark Stokoe and Oli Shelton's paper on how EDM methods can be quantitative in an absolute sense. What metabarcoding is really great and really efficient at doing is detecting spatial and temporal patterns. In other words, changes. And I think there's a lot to be said about understanding changes and patterns. With that, I'm going to conclude my talk and thank you very much for tuning in. If I have time, I'll answer questions. Thank you.
Awesome. Thank you so much, Yuan. We do have time for questions. So if folks want to take a moment to think of some questions and go ahead and type them in the question box. I do want to mention, so Yuan mentioned Oli Shelton's work. So Oli actually gave a presentation on using eDNA to quantify hake abundance. So you can actually go check out the Omics website to see that seminar and a link to that publication as well. So we do have one question for you, Yuan. This one is from Linda. Given that macroalgae are biogenic habitat as well as contributors to the food web, could some of the potential pathogens be important for making kelp bioavailable to consumers?
Catherine, could you repeat the last part? Could they be used what?
Oh yeah, absolutely. Could some of the potential pathogens be important for making kelp bioavailable to consumers?
Pathogens make kelp bioavailable to consumers. I'm not completely sure if I understand the question. Does Linda want to speak up and clarify?
So Linda, if you wouldn't mind, go ahead, and if you want to just go ahead and type in the question box that elaborating on your question, we can get to that in just a moment. Okay, Yuan, I have another question that came in for you while we wait for Linda's clarification.
Sure.
So this one is from Christopher. It's kind of a long one, so maybe we will break this up
Okay.
into a couple of parts. So first of all, he says, great job. I totally agree. Have you considered looking into common wastewater derived viral pathogens such as hepatitis, norovirus, rotavirus, or viral pathogen indicators like male specific choliophage? And then he has like a paragraph here written about that explanation, but I don't know if you want to answer the question first.
Thank you, Christopher, for the question. I personally do not work with the virus. I do understand. I mean, I remember my dissertation work time. I have a lab mate who had to work with the virus, and it's a totally different, quote unquote, animal to handle. You have to concentrate a lot of water. I mean, unfortunately, that is not my specialty, but yeah, you can certainly use with COVID and everything. You can certainly use molecular-based method to track viruses for sure.
Awesome. Thanks, Yuan.
Mhm.
All right. Yeah, I'll go ahead and read Christopher's justification here. He says viruses tend to be in greater abundance and more resilient in the water. During the kelp growing season, viruses also generally have a lower ineffective dose, which could make them more of a public health concern for foods like kelp than some of the bacterial pathogens that often require filter feeding to be present in shellfish at clinically significant levels. But it looks like you touched on that already in your answer. Awesome.
Yeah, mhm, yeah, that's good to know. I actually personally haven't read much about viral pathogens towards kelp. But yeah, thank you for the information.
All right, all right. We got a lot of great questions coming in. So this next one is from Jordan. Sorry if I missed a slide, but do you have conclusions regarding human health concerns of aquaculture kelp from this work? And they follow up by saying, great presentation.
Oh, thank you very much. The quick answer is no. At least, of course, I want to qualify my answer by saying our collection covered only the month of April and May, the major kelp growth season. As you know, what happens after that, we just didn't really take samples. But within April and May, it didn't seem like there were any concerning kelp human pathogens, growing on aquaculture sugar kelp.
Awesome, thanks, Yuan. All right, our next one is from Catherine. She says, thank you, Yuan. Do you have any evidence that, I'm going to pronounce this wrong, so I apologize, Parsu-bacteria?
Bacteria.
Yes, thank you.
Mhm.
Were more prevalent using your brush method, e.g., do you have any evidence that they're endophytic?
Hmm. Great question. Brushing was the only method when I took the field samples in 2018. That's the study year. So I don't really know if I had chosen a different method, what the results would be. Yeah, I do wonder if maybe like some kind of the fluorescence in situ hybridization, like another method, can better identify the location of these bacteria if they were indeed present. But no, it's only surface bacteria that I looked at, and I used only brushing as a collection method.
Great, thank you, Yuan. Okay, we had Linda chime in to clarify her question. So I'll just go ahead and state the original question. She says, given that macroalgae are biogenic habitat as well as contributors to the food web, could some
Mhm.
of the potential pathogens be important for making kelp bioavailable to consumers? And then later she says, some of the potential pathogens could degrade the kelp so it can be eaten by invertebrates and even small fishes.
Oh, okay. So I really appreciate the perspective, not like human-centric. Um, I suppose the answer is yes, because just because it's pathogenic to a human doesn't make it not a good carbon source for other invertebrates to consume as food resources. Again, I will be honest, I haven't really read paper suggesting that, but yeah, I really don't see why it couldn't work that way. I hope that makes sense.
Awesome. Thank you so much, Yuan. It looks like that is the conclusion of our questions. We did have a couple of folks chime in and say thank you for your presentation, that it was a good presentation. So with that, I think we will go ahead and wrap up. If folks have any other lingering questions, feel free to reach directly out to Yuan. You can always email me at noaa.omics@noaa.gov as well, and I'm happy to pass along any information to Yuan as well. So Yuan, thank you so much again for presenting today. We really appreciate it. And huge thanks to our audience for joining us today as well. Our next seminar will take place on October 19th at 12 p.m. Eastern Time. We're going to have Dr. Jean Lim from the University of South Florida, who will be presenting, and her talk is titled Metagenomic Discovery of Microbial and Host Genetic Features of a Marine Polychaete Colonizing a Methane Hydrate in the Gulf of Mexico. So the registration link for that webinar is currently up on the Omics website. So I hope to see you all then. And thank you all so much again for joining, and we'll go ahead and sign off.
Thank you.